So the membrane proteins are individual molecules which are attached to the inner or outer membrane surface. So for the outer membrane surface, okay, we call them peripheral proteins, and for those who are embedded on it is the intrinsic proteins. So we will be taking a look at the figure 2.4 with the next slide. So the arrangement of cholesterol between lipid molecules of our lipid bilayer has a great importance in the stiffening of the cell membrane's phospholipid bilayer. Okay, it stiffens the outer lipid bilayer and causing the inner region of the bilayer to become slightly more fluid. Okay, only half of the lipid bilayer is being shown in the image. The other half is your mirror image. The fluid region has a more unstiffened parts, but the hydrophobic or nonpolar regions have the stiffened parts. So it's because of the cholesterol. And number 4, the structure of the cell membrane. As your carbohydrates are uniting with proteins, they form glycoproteins, which are uniting with lipids. They form glycolipids on your surface of the plasma membrane. Surface carbohydrates and some portions of the proteins and lipids making up the glycocalyx, or we call it cell coat. So the glycocalyx showing the glycoproteins and glycolipids. Note that all of your attached carbohydrates are on the outside of your plasma membrane, and we have protein fibers inside, so it also helps in the facilitation of the substances found in your cell membrane. So what are the functions of your cell membrane? Because it has a very organized structure, so it regulates the material from in and out of your cell, and it separates the inside of the cell from the outside, so to prevent the substances inside your cell from leaking out, and also to separate various organelles within the cell, with providing a large surface area on which the specific chemical reactions can occur, and separate cells from one another. Also, lastly, sites for different receptors containing cell-specific identification markers that differentiate one cell type from the other. That's the purpose of your cell membrane. So what are the movements, okay, that is being regulated by the membranes? Okay, molecules are coming through membranes in a number of ways, both by using their own energy or by relying on energy in an outside source. Okay, so we have the table 2.2 that will discuss the different kinds of a transmembrane movement, and this I will just be focusing on some certain parts. Okay, so for osmosis, this is a type of a movement across the plasma membrane that involves the water molecules. Okay, water molecules are the H2O will diffuse across the selectively permeable membrane. Selectively because it chooses what substances or solute molecules that are coming in from an area of higher concentration to an area of lower concentration. So when you try to examine the frog, okay, the frog's red blood cell, when the concentration of water molecules outside the blood cell is greater than the inside, so that the blood cells of your animals, okay, water will come inside the cell, okay, when there is a greater concentration, okay, outside of your cell, and so on. We have filtration, active transport, endocytosis, which is the bulk movement of your cell by forming a vesicle. Phagocytosis, okay, it's like the drinking, okay, of your cell. And pinocytosis, okay, the droplets of your fluid will take it to the cell. Exocytosis, the movement of the bulk movement of your molecules going out of your cell, like the sciatic nerve of your frog, which releases a chemical neurotransmitter. So we have simple diffusion. Molecules will randomly move at all temperatures above absolute zero, okay, from areas where they're highly concentrated and when they are less concentrated, until they're evenly distributed in a state of dynamic equilibrium, or we say the balance. So the simple diffusion, okay, is being shown on this example, where a sugar cube is being placed in water. It slowly dissolves and disappears. As this happens, the sugar molecules diffuse from a region when they are concentrated to a region. Okay, another C, when they are less concentrated. And lastly, a distribution of your sugar molecules are being distributed in the water, and it reaches the equilibrium. So facilitated diffusion is also a kind of movement where polar molecules not soluble in lipids may diffuse through protein channels, or we have what we call as the pores in the lipid bilayer. So these protein channels are offering a continuous pathway for specific molecules to move across the membrane of your cell, so that they will never come into contact with the hydrophobic layer of the membrane's polar surface, because the membrane of the polar surface is kinda doesn't like water, so these pores are helping it to come in. So the transport proteins, okay, or the protein channel pores, are transporting these molecules from outside the cell, or we call that extracellular fluid, going inside. So facilitated diffusion is involving the large and sum of big molecules, we call it, like glucose and amino acids, which are not soluble in lipids. They require resistance in passing through the membrane. Okay, these molecules use facilitated diffusion, so it doesn't have an energy input like the simple diffusion, but to pass through the membrane, they are binding the carrier or the transport protein found in your cell membrane, and transported from the area of higher concentration to lower concentration. So this one is a carrier protein which fits the circle, okay, shape of your molecules. So once it once it fits the carrier proteins, it will be transported in the cytoplasm of your cell. So also with the osmosis, okay, again, it's a special type of diffusion, not a different method. It involves the movement of water in your area of a higher concentration to an area of lower concentration. So the osmosis here is depicted with the white sugar molecule and the blue water molecule. So this is a selectively permeable membrane, an experiment set up in a beaker. So through time, the water molecules have moved, okay, from 2 to 1. Okay, so once it reaches an osmotic equilibrium, the number of sugar molecules in compartment 1 does not increase, okay, but the number of molecules does increase because they moved in, okay, from 2 to 1 through time. So also, there are some recent studies involving the cell membranes, okay, that are carrying water molecules. So water flow in living cells is facilitated by specialized water channels. We call it aquaporins. Aquaporins are typically has two general classes. We have one specific only for water, and others are small hydrophilic molecules like urea, glycerol, to cross the membrane. So the term tonicity for osmosis is referring to a relative concentration of solutes of the water inside and outside the cell. So for example, we have an iso, which means equal, and tonus, tension. So the solute concentration is the same in inside and outside of your red blood cell. So we will be showing that one later. Hypertonic is the concentration of your solute is higher outside the red blood cell than the inside. Okay, so the result of your hypertonic solution, water will move out of your cell, so it shrinks. So this is what we call as crenation in your red blood cells. And hypotonic, when the solute concentration is lower on the outside of the red blood cell than the inside. So what happens, the water moves out of the cell, okay? It will swell and may burst. So take a look at this effect of your salt concentration on the red cell volumes. Okay, isotonic, normal cells. In hypertonic, they have shriveled cells with crenated, and hypotonic, where the cells will swell and eventually burst. Again, another type of movement is what we call as filtration. Filtration is a process that forces small molecules across selectively permeable membranes with the aid of water pressure or some other externally applied force, such as the blood pressure. So for your frog, filtration is evident when blood forces the water and dissolved molecules through the permeable walls of your small blood vessels called the capillaries. Okay, a specialized blood vessel. Okay, we call the capillaries, which is permeable also. So this is your capillary membrane. We have the smaller molecules that are passing through the capillary membrane and larger molecules that could not go out. Okay, so they are passing through and going to the tissue fluid. So this is the high blood pressure in your capillary forces the small molecules to pass through the capillary membrane. So what is active transport? Active transport is a process that moves molecules across a selectively permeable membrane that is from the area of lower to higher concentration. So it uses ATP or energy to work. So active transport chain is similar to diffusion, except that the carrier protein in the plasma membrane must use energy. Okay, so they have to use energy for them to process. So we have the carrier proteins, the uniporters. Okay, they transport single type of molecule or ion. When they symporters, they transport two molecules or ions in the same direction, and when they antiporters, they could transport molecules or ions in the opposite direction. So this is the example of an active transport. So with the use of energy, this oblong carrier proteins in your cell membrane is transporting the ions from inside outside and vice versa. So bulk transport is for the large molecules. Okay, for bigger molecules, we have endocytosis and exocytosis, which together provide a bulk transport into and out of the cell. So the bulk refers because many substances or molecules are moved at the same time. So in endocytosis, the plasma membrane envelopes the large particles and molecules. Okay, so we have three forms of endocytosis. We have pinocytosis, phagocytosis, and the receptor-mediated endocytosis. For endocytosis and exocytosis, it's responsible for the bulk transport of molecules from into and out of the cell. So take a look at this one: the invagination, formation of pouch, formation of vesicle, and joining of the vesicle with the plasma membrane, and secretion of the cellular product. So these are coming in and coming out of the cell. In pinocytosis, it's a non-specific uptake of the droplets of your fluids found on the outside of your cell. Okay, remember that phago, which means to eat, similar to pinocytosis, except that the cell takes in solid material rather than the liquid. Okay, another thing about the receptor-mediated endocytosis, it involves a specific receptor protein in the membrane that recognizes the extracellular molecule and binds with it. It stimulates the membrane to indent and create a vesicle containing the selected molecule. So we'll take a look into the cytoplasm, organelles, and cellular components of your eukaryotic cell. So again, organelles are the little organs in your cell. So they compartmentalize cell activities. They are enabling the cells to, secrete different substances. Maybe it is enzymes. Okay, they derive energy from nutrients that are being taken by the animal. They degrade debris and waste products and reproduce. So what are the purpose of different structure and function of these organelles are being discussed in more detail. So these are just the components with their structures and their functions. So take your time to read, pause this video and try to read all of these stuff. So we'll start with the cytoplasm. Cytoplasm has two distinct parts. Again, the fluid cytosol suspends all the structures of your cell membrane, and the eukaryotic cell, it contains also dissolved molecules. Okay, this endomembrane system consisting of the ER, or the endoplasmic reticulum, the Golgi apparatus, vacuoles, and vesicles. So for ribosomes, we have protein workbench. Okay, so ribosomes are non-membrane bound structures that are sites of protein synthesis. So they are containing the equal amounts of protein and a special kind of ribonucleic acid. We call that rRNA. They are attaching to the endoplasmic reticulum. What does it mean? So whether they are free floating in your cytoplasm or attached to the ER, they usually cluster in groups connected by a strand of another ribonucleic acid. We call that mRNA. Okay, these clusters are called polyribosomes or polysomes. So the messenger RNA is important in the cell division process or transfer of the DNA going to proteins or amino acids, and we will be discussing that one in cell division and inheritance. The endoplasmic reticulum is a complex membrane-bound labyrinth of flattened sheets, sacs, and tubules that branches and spread out the cytoplasm. So it acts like a railway to your ribosomes. Okay, coming from the nuclear envelope, going to the plasma membrane, it has a series of channels that helps various materials to circulate throughout the cytoplasm. It also stores enzymes and proteins as a point of attachment of your ribosomes. When they are attached with ribosomes, we call them rough ER, and when they are without ribosomes, we call them smooth ER, like this example. So you notice the double membrane and the lumen space within in letter A, and you will notice that the smooth endoplasmic reticulum doesn't have ribosomes. Next are the packaging and sorting organelles of your cell. It was named from Camillo Golgi, which discovered this Golgi apparatus or Golgi complex, which is associated with the ER in the cytoplasm. So the proteins that ribosomes are creating are sealed off in these little packets. We call them transfer vesicles. So transfer vesicles are passing from the ER going to your Golgi apparatus and fuse with it. So they are like this is shown in this image. Okay, it has a stack of cisternae. Notice that the curved cisternae is found in there. It sorts packages, and secretes cell products. Some of the secretory vesicles are moving in the Golgi apparatus from outside, going to the outside of your cell via the exocytosis movement. So lysosomes, again, is a different membrane-bound spherical organelles that releases an acid hydrolase. Okay, they are capable of digesting organic compounds like lipids, proteins, nucleic acids, and polysaccharides under acidic conditions. So these enzymes are made in the ER and then transported to your Golgi apparatus for processing, and they are secreted by the Golgi bodies in the form of lysosomes or vesicles that fuse with lysosomes. So they are the suicide bags, okay, of your cell. So they remove the waste products of your cell. Oh, so you can see it here in the image of your lysosome formation. They are arising from the Golgi bodies. Try to see the yellow arrow. And fuse with the vesicles that engulf foreign material to form digestive vesicles. We call them phagolysosomes. Okay, these vesicles are functioning in the normal recycling of cell constituents. So we have also microbodies, a diverse category of organelles. So enzyme-bearing membrane-enclosed vesicles are called microbodies. So the enzymes into microbodies is one of the main way where the eukaryotic cells were able to organize their metabolism. So one type of a microbodies we call that peroxisome. So they also contain enzymes that catalyzes the removal of electrons and associated hydrogen atoms. For example, hydrogen peroxide. So we have peroxisomes. So mitochondria, or for mitochondrion, are also a double membrane bound organelles that are spherical to elongated in shape. So they have these particular partitions or folds. We call that cristae. You will see here a mitochondrion. It has a membrane, it a cristae and matrix. Also, it contains DNA, ribosomes, and enzymes. So we call that mitochondrial DNA, or the mtDNA that comes from the maternal side of the animal. For cytoskeleton, it's easy to remember because it's the cell skeleton of your cell. Okay, so it forms a lattice framework that extends throughout the cytoplasm and connecting different organelles and cellular components. It makes the cell its shape and its scaffold. So this is the model of your cytoskeleton, showing the 3D arrangement of the microtubules, intermediate filaments, and the microfilaments. So what are microtubules? Okay, these are hollow, slender, cylindrical structures of your animal cell. So they are made up of subunits called the tubulin. Okay, subunits. So they are moving. They help the organelles to function in the movement. Okay, we have the secretory vesicles and the chromosome development during the division of cell's nucleus. So also they help in the transport system of your cell, kasi there's a lot of transport that's happening in the cell, like your nerve cells. So they have helped move materials through the long nerve processes, and they are an important part of your cytoskeleton, the cytoplasm, because they change the shape of your cell, which is very important when they are undergoing different types of specialization. So they need to adjust the shape. So we have intermediate filaments. So these are chemically heterogeneous group of protein fibers. Okay, so these have specific proteins which can be carried by different cell types. So, and these filaments are maintaining the cell shape and spatial organization of organelles. The same thing with mechanical activities of your cytoplasm. And we have actin filaments on solid strings of proteins. We call it actin molecules. Okay, they are highly developed in your muscle cells. We call that myofibrils. They allow your muscle cells to shorten or contract. Okay, actin microfilaments in non-muscle cells provide mechanical support, just like the contractile systems of your cellular movements in amoeboid movement in some protozoans. And these are the three major classes of protein fibers making up the cytoskeleton of your eukaryotic cell: the microtubule, the intermediate filament, and the actin subunits. So we now go with the cilia and flagella for movement. Okay, cilium is, or the cilia is coming from the Latin word eyelashes, and flagella, or what we call as the flagellum, small whips. Okay, they are the elongated appendage of the surface of your cell, eukaryotic cell. Also with the prokaryotic cell. Some of them have cilia and flagella, but in stationary cells, cilia or flagella move material over the cell's surface. So the propulsion, the locomotion is being used with these two organelles. So although flagella are 5 to 20 times as strong as cilia, they move somewhat differently. So cilia and flagella have almost the same structure. Both are membrane-bound cylinders that enclose a matrix. We call that axoneme or axial filament, and they consist of 9 pairs of microtubules arranged in a circle around 2 central tubules. For the cytoplasm of the base of its cilium or flagellum, there is what we call as the cylindrical basal body. So they are made up of microtubules and they are structurally identical to the centrioles. So they are having the 910 pattern, 9 sets of a tree with none in the middle. So this is the internal structure of your cilia and flagella in cross section. So we have here the dinane arms and a tubulin subunits, and we have the basal body and the spoke with a plasma membrane with also a microtubule that comes into the doublet. So centrioles and microtubule organizing centers are specialized for the non-membranous region of your cytoplasm, because these dense materials will give rise to a large number of microtubules with different functions in the cytoskeleton. Okay, so for instance, one type of center gives rise to the centrioles that lie at right angles at each other. So this is very important organizing centers of your eukaryotic cell. For cell maintenance, we have vacuoles. Okay, they are membranous sacs that are part of the cytomembrane system. So vacuoles, you noticed earlier that they come in different shapes and sizes. Also, they have various functions. For example, for protozoans and sponges, they have contractile vacuoles that collect the water and pump to the outside to maintain the organism's internal environment. Other vacuoles can be storing food. Others can be transporting ions. These are volts. Okay, sometimes they are referred to the mysterious symmetrical cells. So these octagonal barrels are cytoplasmic ribonucleoproteins. Okay, their function may be related to their octagonal shape, and one cell may have thousands of volts. Okay, they are similarly, the nuclear pores are also octagonally shaped. The same with volts. They lead to speculation that volts may be cellular tracks. So these volts are under investigation. Take a look into that structure of your volts, a 3D drawing. So what are exosomes? Exosomes are being discovered just recently. Okay, so that's a good thing about science. It evolves constantly. It accepts change. For scientists to discover new stuff for this decade or next decade, it's really interesting that there is some new background knowledge relating cells, organelles are being found. Okay, so however, these exosomes are considered little more than garbage. Okay, garbage cans that are to discard the unwanted cellular components. So we have evidences that are showing that accumulated dumpsters also act as messengers. Okay, they convey information to the other cells and tissues. So these secreted vesicles are the signaling structures of your cell. So we have the nucleus as the information center. So this is the more in-depth explanation of the nucleus, which contains the DNA, and it controls the information center of the eukaryotic cell. So what are the functions of your nucleus? Okay, it directs chemical reactions in cells by transcribing genetic information from DNA into RNA, into amino acid, okay, to proteins. So there are some specific cell activities which is needed by the cell, so nucleus has to act on it. It also stores genetic information and transfers this information during cell division from one cell to the next and from one generations of organisms to the next. The nuclear envelope is a membrane, okay, that separates again from the nucleus from the cytoplasm, and it's continuous with the endoplasmic reticulum at a number of points. As I said earlier, they are connected with the nucleus because typically your nucleus has 3,000, more than that penetrate the surface of your nuclear envelope. This example an electron micrograph of a section through the nuclear envelope showing nuclear pores or arrows. So what are the genetic containers of your cell? We call them chromosomes. Okay, so nucleoplasm is the inner mass of your nucleus, and it contains the chromatin, which is a genetic material. So the chromatin are consisting of the DNA, proteins, and the uncoiled tangled mass. We call them chromosomes, or the colored bodies, because the scientists once discovered them under the microscope, and they have this colored body, so we call them chromosomes. They contain DNA that houses the genes, and during the cell division, the chromosomes coils tightly, making the chromosome visible when viewed in a light microscope. Nucleolus are also a part of a nucleus, which is a non-membrane bound structure in the nucleoplasm, which is present in your non-dividing cells. Okay, so for your amphibian cells, we have 2 to 3 nucleoli form in most cells, but also they could be in thousands. So for amphibian eggs, they release more nucleolus. Okay. These are the three some major structures of your nucleus: chromatin, nuclear envelope, and then nucleolus in your light microscope in a 7,900 magnification. So what are the level of organization in various animals? So we have the first level, we call that protoplasmic organization. Okay, for protozoans that are unicellular, okay, we call them protoplasmic, okay, because they are confined in a structure of a single cell. So a second level, we call them cellular organization, like the flagellates, such as the volvox and some sponges, can be placed at this level, because we need an aggregate of cells to call that cellular organization. Okay, they are differentiated. They exhibit a division of labor among these cells. And the third level is the tissue level, which we belong, mammals, okay, and vertebrates. So jellyfishes have aggregations of cells organized into a definite patterns of layers which form a tissue. And the fourth level is the organ level. Okay, organs are composed of one or more tissues and have more specialized functions than tissues. The level first appears in flatworms, where the specific structures such as reproductive organs, eye spots, and feeding structures are present. So the fifth and the largest level of organization is a system level. So again, tissue level is applicable for jellyfishes, okay, and lastly, the system level is applicable for vertebrates. Okay, apologies for having confusion. So the tissue is an aggregate of cells. Okay, again, histology is a study of tissues, and they are classified into 3 different type of tissues: the epithelial, connective, muscular, and nervous. So they perform a certain function, and the first one is the epithelial tissue. So it has many different forms. The most typical function of your epithelial tissue is that they are basically differentiated according to their function. Okay, a general idea of the epithelial tissue is that they contain a basement membrane. Okay, they have separating the epithelial tissues from the underlying adjacent tissues. So they absorb, like the lining of your small intestine, they transport nutrients, waste products, excretion, and protect your skin. And also they contain nerve cells for sensory reception, just like your taste buds in your tongue. Okay. We have also differentiated in the basis of shape and number of layers present. So we will be taking a look at these images. So the tissue type of this one is a simple squamous epithelium. They are found in a air sacs of your lungs, kidney glomeruli. So what are the functions of this? They allow the passage of materials by diffusion and filtration. They are somehow simple and flattened. This simple cuboidal epithelium are tightly packed, and they are having the cube-shaped cells. So they are found in your kidney tubules, small glands, and the surface of the ovary. So most of their function are for secreting and absorption. Simple columnar epithelium is consisting a layer of elongated cells. So take a look at their sizes and shape. They're more of a narrow type of epithelia. They line the digestive tract, the gallbladder, and the excretory ducts of some glands. So what are their functions? Absorption and enzyme secretion. We have the pseudostratified ciliated columnar epithelium. So try to look at the cilia here on the surface. So it lines your bronchi, uterine tubes, and some regions of your uterus. And what are the functions of these pseudostratified ciliated columnar epithelium? Is they propel mucus or reproductive cells by cilia reaction. So these cilia are like moving the stuff like mucus or reproductive cells. We have stratified squamous epithelium that are lining your esophagus, mouth, and the vagina, and some of them can be keratinized, okay, and lining the surface of your skin, and non-keratinized when they are found in this like vagina. They protect the underlying tissues in areas that are subject to abrasion. For connective tissues, we have the support and bind, okay? They are distributed through the extracellular matrix, and this matrix frequently contains fibers that are embedded in a ground substance and with a consistency from a liquid to solid. So these are the properties of various connective tissue from the root word to connect, okay, and provide support. So do the general types of fiber arrangement in loose connective tissue. We have strong, flexible fibers of protein collagen, which are interwoven, and reticular fibers giving loose connective tissue its elastic consistency and making it an excellent binding tissue. Like this one. Fibroblasts are found in here in arrows, like this one. Okay, they are suspended with in a gel-like substance. Okay, so these are found in the epithelia, under the epithelia of your human body, and it wraps your different organs and cushions your organs. Collagenous and elastic fibers are also found in these black stripes. So fibrous connective tissue, the most obvious fibers are called collagen fibers. They are densely packed and they lie parallel to one another. So they are